some of the preliminary results from a comparative phylogeographic study that I'm doing in the Central African countries of Gabon, Cameroon and Equatorial Guinea to try and explain the historical processes that have been responsible for generating biodiversity within species. So there have been a kind of sparse handful of studies on various species which have suggested that three different historical processes have played a role, so in generating diversity within species. So firstly, a kind of traditional Pleistocene forest refugia model. Secondly, that river barriers have been important in constraining dispersal within species. And thirdly, a seasonal inversion effect across the equator, which I'm going to explain in a bit more detail in a further slide. So there haven't been any attempts to kind of bring together data from different species to analyse it under a kind of comparative statistical framework. So this is what I'm aiming to do in this study, and I'm using mitochondrial DNA, which is kind of widely available for a number of different species in the region. So I'm using a program called Phylogeosim, which is a phylogeographic simulator, and it's a program which enables you to firstly simulate demographic history through time under whatever hypothesis you're testing. Then, under that model of demographic history, go back in time using coalescent theory to simulate genetic data. So genetic data under that model of demographic history. Then if you simulate, say, 1,000 genetic data sets under each model of demographic history, you can see how well your real data set falls within your simulated data sets using summary statistics. And this program also enables you to define parameters that relate to species-specific ecology, such as effective population size, short and long distance migration. So then you can really tease apart the effects of demographic history and species-specific ecology. So in these diagrams, this shows the kind of models that I'm testing. So Central Africa has been divided into a number of cells. Where the cell is coloured green, that means it has a maximum effective population size, so it contains good habitat for the species. If it's coloured in blue, that means that it's water, so it has an effective population size of 0. And if it's coloured in white, that means that it has habitat that's not suitable, so it has an effective population size of 0 too. And where the cell is coloured in yellow, that means it has restricted dispersal in this area. And the red cells are those that have been sampled. So when we simulate genetic data, we will have genetic data simulated from those populations. So there are three different scenarios. The first is one of Pleistocene refugia. So basically, during the glacial periods of the Pleistocene, the populations were constrained to these small isolated refugia and then kind of diverged in allopatry, and then expanded to fill the entire region after the last glacial maximum. The second scenario is one where these two big river basins were kind of restricting dispersal. And the third is where there was reduced migration across the equator. And basically, due to the kind of equator effect, the dry seasons in this region, south of the equator correspond to the rainy seasons north of the equator. So you kind of have a seasonal inversion effect that takes place over quite a short geographic distance. So I'm comparing, and also you can combine these models. So you can have, for example, Pleistocene refugia with a river effect, Pleistocene refugia with an equator effect, etc. So I'm simulating genetic data under each of these scenarios and comparing it to real data from a number of different species. 2 different rodent species, 2 duiker species, which are small forest antelope, 1 ape species, and an amphibian too. And some preliminary results. I just have results so far from 2 different species, 1 mouse species and 1 duiker species. And so far, in both species, a model of refugia is supported, so it looks as if contraction to smaller refugial areas during the Pleistocene has played an important role in shaping genetic structure in these species. In the mice, there's just 1 refugium supported versus 2 refugia in the duikers. Rivers in the mice do improve the model, whereas they don't in the duikers. So it looks like duikers have historically been better at dispersing across rivers, basically, which kind of makes sense. Also, the mice have comparatively a high effective population size, where they have a higher population density and lower migration rates. So this kind of convinces us that our model is working. And we're going on to compare the real data from other